Hello and welcome. My name is Mitch Sanders and he is Kyle Quinn. And we're delighted to host another Wound Healing Society Fireside Chat. We have two very special guests, Luisa DiPietro, who's the professor, UIC Scientific Director for Chicago Biomedical Consortium, the Director for UIC Center for Wound Healing and Tissue Regeneration and Periodontics. And we also have Trevor Leonardo, who is a MD PhD, yes?
Yep.
Wonderful. And you're at U Chicago in the microbiology immunology department, correct?
Yep.
Yep.
Welcome.
University of Illinois, Chicago.
I love Chicago. I'm actually going out for the Guster conference. Not Guster Guster concert. It's a
Really?
band.
I know who they are, yes.
Okay. It's a band from Tufts. It goes
Yeah.
way back when.
Yeah, cool.
So today we have a wonderful paper that we're featuring, transcriptional changes in human palate and skin healing. And before we get started, Luisa and Trevor, can you provide something to provide your kind of background, your story of how you got into wound healing in the first place?
Yeah, I'm going to talk a little bit about how we got into, oh, you mean my background of how I got interested in wound?
Yes, yes.
Yeah, so for me, what happened was I was in grad school and I was actually studying immunoglobulin genetics at the time when molecular biology was booming. But I was doing a lot of reading about immune cells, including phagocytes, and I learned that phagocytes have a lot to do with healing and repair, and that really appealed to me. Because I could see the intersection of immunology then with something very clinically relevant to my clinical background in dentistry. So luckily for me, there were two people at Northwestern, Joe Leibovich and Peter Polverini, who were really interested in macrophages as mediators of growth, particularly actually in tumors. And I went down there and worked with them. And after I finished my PhD and sort of changed my whole life around and became a wound healing person. And I never left the field because it's just such a fascinating field and I love working in it. And maybe Trevor, you want to say how you got here?
Yeah, so during my undergraduate years and also after finishing my undergraduate degree, I was doing a lot of stem cell research and looking at the regenerative possibilities and capabilities of those types of cells was really interesting. And then when I was a second year medical student, we were learning about pathology and how inflammation occurs in almost every setting or every pathology. And it really got me thinking about how, if we could better understand how the body responds to injury or inflammation responses in different tissues, and this fibrotic response, that's kind of a common theme across all these tissues, maybe we could better understand an approach to address pathologies on a broader level. And that led me down to the wound healing and tissue regeneration focus with Dr. DiPietro.
I think the paper is outstanding. I'm gonna let Kyle ask the questions. I think for the viewers of this video, you should really dig deep into the paper and look at the supplementary figures because there's so much. It's very rich in data, and some people forget to look at the supplementary figures, and they're just beautiful. So, and I'm really excited about the different cells that you're seeing in the different tissues and how that may be playing a role in the genomics. So. Kyle, why don't you take it away and I'll join in it when I can.
Sure. Yeah. I mean, yeah, very interesting paper, comparing the oral mucosa in our skin. And I guess could you start by telling us the motivation for this study?
Right. So I'll take that question. We had been looking at the differences between oral mucosal and skin repair for a long time because, it's been clinically noted for a very long time. Clinicians have observed it, oral surgeons, ENT, people who work around the mouth. And so that there was this clinical perception that the oral mucosa healed better, more rapidly, and with less scarring. And it was known, of course, that you don't get keloids, for example, in the oral cavity. So there's something different about it. But we started to study it a long time ago, and we had this mouse model, but the oral mucosal part of that model was the tongue, because that was the easy place to wound. And we published several papers on that, just sort of just describing the differences in angiogenesis and scar formation and inflammation, which is less. But we really wanted to do two things: move towards human, and then also move towards something that might have a more direct histologic comparison between skin and mucosa. So for this study that Trevor is working on, or worked on, he's still working on, he's going to tell you about it, we decided to use the palate. Because when you compare the palate to the skin, so the hard palate to the skin, it's very similar histologically. The hard palate has keratinized stratified squamous epithelium. It has a connective tissue underneath that looks a lot like dermis, but it's called the lamina propria. And as compared to tongue, it doesn't have all those extra things like taste buds and follicular situation where you have to worry about all those extra tissues. And also the tongue has a lot of muscle underneath it, which you don't find in the palate. So the palate was really ideal. And we also wanted to move to human. So this is a human study. And that was a bit of a challenge, but we were able to get our IRB to approve these paired human mucosal and skin wounds. So that was really the reason. And then the goal was to look more globally. So instead of looking just at individual cells or factors, to look at really the genomic response.
Yeah, great. So I guess kind of digging into, you mentioned, this is obviously a human subjects study. Can you give us just an overview of the study design, and then we can kind of get into also some of the analysis methods that you employ, because there's a lot there.
I'll tell you about how the samples were obtained, and I think Trevor's going to take over for the analysis. But the samples were obtained quite a long time ago, actually, by Phil Marucha. And they're paired samples on volunteers. The volunteers are young. They're like 18 to, I think, 35. And so we chose a population of fairly restricted age, so they would all be similar in that sense. And then there's paired wounds that are placed either on the palate in a very specific place related to where the molars are, and they were 1 by 5 millimeters in size and about 1.5 millimeters in depth. And then a similar size wound was placed on the upper inner arm. And then at various time points, those wounds were completely biopsied with a 2 by 5 excisional wound to really capture all of the wounded tissue. So that's how it was done. And I forget, actually, Trevor, you may remember how many subjects we had and how many samples.
Yeah, so I guess I can step in at this point. So we had 18 patients total, 9 of male and 9 female. There were about 96 samples total in terms of like every time point that was collected. This again was in 2 tissues, so the palate and the skin, and there were 5 time points that were collected. And so moving forward, we can kind of build this framework where we have 0 hours representing our unwounded tissue, or the initial collection of the tissue. And then every subsequent time point is 6 hours, day 1. So those are the more acute time points in terms of the wound healing response. And then also day 3 and day 7. And we don't have samples collected from every patient at every time point, because that would have not worked in terms of, for a bunch of logistical reasons and IRB reasons. But on the paper in our first figure, actually, it shows which samples were collected from which patient at which time point. So yeah, that's basically the study design. Do you want me to talk about the analysis methods now?
Sure. I've got just a quick follow-up question, just out of curiosity. You mentioned that these were taken in a very specific location in the palate, relative to the molars. What was the rationale for that particular location?
Oh, nothing really, except we wanted to be uniform. So we used the first molar as a marker, just so we'd always be in the same location. Because a hard palate, it's pretty big up there. And actually, interestingly, there was just a paper that came out that says in the mouse, the anterior palate heals differently than the posterior palate. So I'm kind of glad we stuck to the one location.
Yeah.
At the time, we didn't realize that, and certainly we hadn't looked at that in humans. But so I'm not sure if it's the same, but that has been now described in mouse.
Yeah, so let's get into the analysis methods.
Yeah, so this data was like the tissues were collected and then processed, pretty standard approach in terms of like RNA isolation, and then they were run on an Affymetrix array. There was a quality control analysis done that's pretty uniform across like most methods that we used. And then we used a brain array mapping technique where since this data was, these Affymetrix chips were created so long ago, we wanted to make sure that all of these probes that were pre-designed back then were actually accurate in terms of the most modern annotation to the genome. So Brain Array is this really cool website that this group has put together where they take the newer genome, genomic information and overlay these probes from these Affymetrix arrays onto the newest up-to-date genome, to where the genes that you're saying are mapping, or the probes that you're saying are mapping to a specific gene actually map to the gene. And so that was like one big step that I think allowed us to use old microarray technology onto modern day genomic information. And then once we had mapped these genes, we did a normal robust multi-chip averaging or RMA normalization that's very common for microarray data. And that gave us about 18,000 annotated genes. We then used Lemma, which is it stands for linear models for microarray data. It's a standard microarray package in R, where you can do differential expression analyses. And so that was our goal, was to compare all of these different samples from these different groups and tissues to each other using a differential expression analysis.
And we quickly learned that it's quite difficult to do this type of analysis over time. A lot of people are simply comparing two conditions. They're not comparing two conditions over time. So Trevor really had to dig in to figure out how to do that. And Dr. Glass, of course, was extremely helpful.
Yeah, and I can go a little more deeply into the study design in terms of why we decided to do the comparisons we did, if you'd like.
Sure, yeah.
So one approach you can take to doing a time series analysis is to actually just have all of your samples in each group be compared in terms of the trend over time. So you could see genes that have differences in their trends or the slopes that change over time. And that's really interesting in looking at like the temporal relationships. And then you could, do a downstream analysis where you see or a clustering analysis or something to see, this group of genes is upregulated over time or downregulated and then goes back up to baseline, et cetera. And so you could do that with like the two different tissue groups. But what I wanted to do, and after talking with Dr. Glass and Dr. DiPietro about this, was first to compare every time point after injury to the unwounded respective tissue. And this does two different things. One, it allows you to control for baseline tissue specific expression and what changes occur specific to that time point. And then the second thing is that when you want to compare across tissues, you now can look at those time point specific changes between palate and skin. Versus if you just did the trends, you could find a lot of genes that would be statistically different over time because you're wounding the tissue. And it's a lot harder to do the downstream analyses after this differential expression, which include like our gene set enrichment analysis, for example.
Yeah, so maybe we can talk about what are some of the biggest notable differences among the two tissues in terms of gene sets.
Can I just jump in? I just wanted to mention
Oh, sure.
one thing. Trevor said something about using the older microarray technology, which we did, and you might wonder why. But that's really because these samples were processed. RNA-seq was really widely available. It really wasn't even financially possible for us to even consider that at the time. But I think the data is still, as Trevor said, extremely valuable, and it's valid. So you know, I don't know why everyone's jumped the other direction. There's a lot of advantages to RNA-seq, but I think microarray still has a lot of, it gives us a lot of great information. Now
We-
Trevor's going to tell you what that info is.
We actually do a lot of nanostring in our lab. And it allows,
Yeah.
you know, the benefit of there is we've made a custom wound care panel of about 300 genes, and it tries to cover everything from macrophage plasticity to fibrosis to scarring and the different phases of wound healing. But please go ahead and tell us about the data and the differences.
Yeah, so just briefly, so our comparisons were every tissue's time point, every specific time point after injury, so that was 6 hours, day 1, day 3, and day 7, was individually compared to their respective unwounded tissue. And so using lemma, you get a modified t statistic, which you can think of for simplicity's sake as a test statistic. And so for all of the 18,000 genes, we ranked the genes from the most positive test statistic to the most negative. And the ones on the each end are respectively like more statistically significantly either upregulated or downregulated. It doesn't directly correlate with log fold change, but it has relationship with log fold change and also statistical significance. And so by ranking all of these genes, we can take this list for every time point after injury versus unwounded tissue and put it into the gene set enrichment analysis, which then will find gene sets or biological processes where sets of genes will be either at the high end of this ranked list or the lower end, representing genes that were statistically significantly upregulated in a process or downregulated in a process. And so that was another thing with doing this time point specific analysis in terms of the experimental design. So the things that we found, first, we wanted to look at overlap between the two tissues. So which processes or gene sets were overlapping in both tissues. And so in one of our figures, it shows, you know, the epithelial to mesenchyme transition is relatively upregulated over almost every time point. There's some immune processes like TNF alpha signaling, other common things you would expect like extracellular matrix organization, and also some general inflammatory responses. We then looked at specific gene sets that were upregulated at multiple time points in either uniquely to the palate or the skin. And with that, we found that the palate had more processes related to endothelium development or cellular response to VEGF, for example, and like sprouting and angiogenesis, which kind of makes sense based on a paper that Dr. DiPietro published earlier back in 2003, where it showed that the the vascular density of the oral mucosa is significantly higher than in the skin. So it makes sense based on the cellular makeup that this would happen or that this would show up. And then in terms of the skin, things that happened over time in multiple time points were like dendritic cell migration, some chemotaxis of leukocytes, and some other inflammatory processes. And then some stuff that's specific to skin, like hemidesmosome assembly. So I think this was showing up at not at the 6-hour time point, which makes sense because that's right after injury, but then reestablishing that barrier that's so important to protecting us from these invading organisms in the skin shows up at multiple time points, I think, as soon as day 1 and every day after.
And hair follicle development shows up in the skin and things didn't show up in the mucosa because that would have been really a problem.
Good sanity check, yeah.
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 I was
Yeah.
really interested in the different cell types. You know, dendritic cells seem to be more prevalent in the skin, as you mentioned, and some of the other cell types that seem more prevalent in the palate. Can you talk about that?
Yeah, so if we jump forward to the cell type enrichment analysis, this was where we took our, we averaged all the samples within a tissue and within a specific time point, their expression, and input that into our, sorry, we did it for every single sample, I apologize. But basically we grouped those after to then see, you know, the respective cell proportions using this cool algorithm called XCell. It kind of deconvolutes a like your tissue data into specific cell type signatures. And the data we get back is first saying whether or not we can detect these cell types of the 64 different cell types that they start with in your tissues at different time points. And then the proportion of those cells with respect to the rest of the 64 cell types. And so one thing we found was that in the skin, like you were saying, there were more dendritic cells and mast cells in the skin and in the palate. I will note that this is not totally statistically valid in a sense, because this is more because of the way that the proportionalities are like generated. But it is really interesting to look at like the trends that you see and also the presence or absence of these different cell types. In terms of other cell types that I think showed up in the skin and not the palate, or more so, were some natural killer T cells and some mesenchymal stem cells, which makes sense to me as well based on the histological makeup. And again, you see like more endothelial cells in the palate versus in the skin. One thing that we also noticed was that there was a neutrophil bump, you know, in the acute response time points in both tissues, which makes sense. More so sustained a bit in the skin versus in the palate. And this was also seen in the macrophages. And this also correlates again with a paper that Dr. DiPietro published, where her and some lab members were quantifying the different neutrophil levels over time in oral versus skin cells along with macrophages.
Yeah, it was really nice that the Excel analysis matched what we'd seen before for neutrophils and macrophages, and also, as we published in this paper, for mast cells. But I think being able to look at all these other cells and now feeling like this probably is accurate for the other cell types, I guess that leads to a future direction, you know, possibly looking at some of these other large differences that we don't exactly understand and trying to find out make sure they're real, you know, histologically, of course, but I bet they are, based on the fact that the others have matched what we've looked at. But, you know, that's something we'd have to do going forward.
And Trevor, I think in the paper it mentions that the early stages of wound healing, the very early kind of inflammatory phase, very similar between the skin and the palate, right? And then when you got to the later days, that's when the palate really showed differences than from the skin. It was remarkable how there was less differences in the palate on the later days. There were very few gene differences. You start out with a huge burst of upregulation and downregulation of a whole bunch of genes, and then later at the end, it seemed like it was really calming down to a set of genes, right? That were.
Yeah, so you're right on multiple accounts. So like in terms of the quantity of genes that were differentially expressed, the palate had a large number of genes in the beginning, and then by day 3 and day 7, it had drastically shifted down to like very few, couple hundred genes versus thousands in the initial time points. Whereas in the skin, it was kind of more sustained across multiple time points, still more in the acute phase, but compared to the palate, like significantly more.
And Luisa, what were there certain types of genes that kind of lead you to believe that you may have good biomarkers or targets for the differences between skin and palate? So you could say, shoot, we should look at this pathway or these groupings of genes to see if we could reduce scarring in the skin.
Sure. Well, this analysis has led us to another analysis, which Trevor's writing the paper right now, so he can tell you about it. But it addresses exactly that point. And that is, are there networks of genes that are moving together, and can we find a target regulator? Because it's not just one element, and all of us know that when you just try to change one thing in wounds, you might get some effect, but it almost never works in a human. So now, Trevor, I'd be happy to turn it over to you, but he's looking to, you know, are there certain regulators that we could pick out that would regulate networks that we see are changing? And in that way, we might be able to functionally improve healing in the skin, for example, by targeting a network of things, not just a single element. So maybe Trevor, you want to expand a little bit?
Yeah, so one interesting thing about this study is, we're looking at the transcriptional responses, not only quantitatively in terms of the number of genes and how they change over time, the degree of overlap between the two tissues at different time points, like identifying a conserved response versus a more unique response, which I think is occurring. But also, instead of just looking at the gene expression changes, and tying those to functionally relevant or biologically meaningful processes, one thing that I was thinking about kind of early on and talking with Dr. DiPietro about in my PhD work was to, instead of identify just these biological processes and the temporal relationships, what if we could identify factors, transcription factors, that are actually causing all of these transcriptional changes to occur over time. And instead of looking specifically at expression, looking at network changes in terms of transcription factors targeting different sets of genes that are biologically relevant. So then instead of maybe focusing on a pathway, we could identify transcription factors that could be leading to a more minimal scarring response versus a scarring response. And also like conserved stuff as well.
So that analysis was done with help of Dr. Glass, who's developed several different methodologies that allow you to look at networks of genes that by prior information are linked by transcriptional factor activity. And so Trevor was able to analyze data and look for network movement within the two tissues. And stay tuned because he's writing the paper and we hope to get it submitted by this fall.
Wonderful. I hope to be a reviewer.
Thank you. We'll keep that in mind.
No, I love the work. I think we're running out of time. I think we've actually had a wonderful time. Do you have any plans are you going to Innovations this year? Or do you have any other conferences coming up? Like, I know there's a Gordon conference coming up in a couple of weeks, right?
Yes, I was at the Gordon, the GRC for tissue repair and regeneration in June, and now I'm going to the one on epidermal barrier function in just a few weeks. So that's my next meeting.
Kyle, are you going to that one? I may send a student, not a research associate, and I might show up and sponsor something. I don't even know I'll just bring a bag of money and give it to them. I don't know.
I'm sure you'd be very welcome in that case.
Yeah.
Yeah, no, I'm not going to that one. But yeah, I haven't been to that particular Gordon. I've been to the tissue repair and regeneration a few times, and I always like that one. But yeah, I'd like to check out the barrier function one at some point.
Yeah, I do think that this is going to help us understand wound recurrence. So that's why I want to be at that meeting. So I think it's a long meeting though. I may only pick a day or two. I don't know if I can go to the whole thing. But anyway, wonderful paper, delighted, great, fantastic work.
Thank you so much for inviting us. I know we really enjoyed being here. And, from my perspective also, it's really so wonderful to have a graduate student like Trevor who's been able to accomplish so much. And as you can see, he's on his way to a fantastic career. So hope to keep him in the wound healing community.
No, it's exciting. I hope you get some awards this year for some of the, we give out a lot of innovation awards and hopefully we'll get to feature you again on your next paper. Anyway, take care. Have a wonderful day. I'm headed back into the preclinical site.
Bye
Thank you so much.
Bye.
Bye.